Hello, my name is Alexandre Brevern, and today I will present our poster, Contribution of Bioinformatics to Blood Transfusion, Database and 3D Intraprotein Interaction Studies, a work done with Aline Flock and Stefan Teletscher. Nowadays, we can consider that we know 43 blood group systems. They consist in 345 different antigens. These figures summarize different types of topology of proteins or sugar associated to this blood group system. After the ABO, the rhesus is the most problematic one. It's also the most complex one, as we will see. In terms of genetics, RH system is encoded by two paralogous genes, RHD and RHCE. They share 92% of sequence identity for the amino acid, and it's higher for nucleotide sequence. They encode for two proteins, RHD and RHCE, and are associated to 5 main antigens. We are interested into the antigens D of RHD. Our collaboration began with a fourth guy, Christophe Tonamil, who asked us what is the effect of a punctual mutation on a RH protein. What happens in fact in 3D? So, using two structural templates with 30% of sequence identity with the RHD protein, we generate a multiple sequence alignment, and with this structural information, we generate more than 100 structural models. Using our own approach called HPM, we have selected at the end the most interesting RHD structural models represented here on this picture. This model was interesting because it can explain what happens with two variants. On the left, you have the position 223 with the wild type phenylalanine. This residue has a lot of interaction with its neighborhood residues and with a lot of other helices. The two other variants associated with transfusion issues have lost near all these interactions. So our hypothesis is that you have a potential protein destabilization that provides new epitopes. To go further, we know that RH is not a solo player, it's in a trimer, so we have tried to propose a trimer structural models. For that, we have modeled different trimers, and here we show you the most interesting one, RHD with two RHAG, a protein that is from the same family and that is supposed to be associated to our protein of interest in red. Like in the previous work, we have analyzed the difference of interaction that we can see for the wild type and the variants. With a little bit less than 20 different variants that can be categorized as important where you have a lot of immunosuppression risk and others that are frequent but with weak impact in terms of transfusion. We have modelized these punctual variants using Squirrel. Here you have the result on this plot. On the x-axis, you have the number of interactions that have been modified between the reference and the new variants in terms of interactions, going to 20 at the end. And on the y-axis, you have the number of helices that have been also modified in terms of interaction. So I repeat, in green, it's frequent variants, polymorphism, that are not associated to transfusional events. And here, with the black cycle, that's at the opposite, important imaginational risk variance. So we have a nice separations here in terms of number of modified interactions. With this work, we had a better definition of the extracellular domains of RHD. We have a nice correlation for the non-immunized variants and for the variants frequently associated with immunization events. The correlation was interesting. When we were doing this work, we were very surprised that to have access, in fact, to clean, simple data on RH variants. We have so decided to develop a specific database, RHeference. For that, Dr. Aline Flock reviewed more than 5,000 articles, conference abstract to see exactly what was the information inside, creating some kind of CSV tables. Here you have the number of information of these tables that have been converted into a BioDjango database. It's now available for the scientific community. We have more than 3 times more entries than other database, but this one you can query it, which is not the case for others. You have information for the alleles, the papers that you can have, documentation, of course, about the database, whole statistics that are quite interesting. And here we have, of course, the URL and a very simple page, a result. Here for the RHD DAO5, we have decided not to provide directly all the information. Users click on the information that you would like to have because there is a lot of information associated to many variants. So the conclusion of this work is that it was a very fruitful collaboration between structural bioinformaticians and medical doctors. Interestingly, we have seen that we can change the view of proteins analyzed since decades by specialists. There is a potential link between polymorphism and transfusion complications through these 3D structural models. And from all these discussions we had, we have proposed a new RHeference database. So thank you. And thank you to have listened to me and to look at this poster.